I'm gonna play the song on loop. I don't give a shit. This song is so good. Normal and mutant cell cycle stimulating pathway. Alright, so Ross. %30 of human cancer? That seemed kind of high. I mean, that's insane. Let's turn this way down. I guess it's getting caught up in it. I don't know what to do about that. I'm not wearing headphones. You couldn't pay me to wear headphones. So Ross is a G protein. So Ross is a G protein. And I don't remember G proteins. G proteins they're in signal transduction. Bro, I should buy a DJ set. Actually, that'd be so fun though. It would take so much time to learn, but like, how much is a DJ set? Honestly, I don't want a DJ set. It's gonna take like. No. Man, the thing is, I don't have Ableton on this computer. Ooh. Yeah, you can buy them for like $150. Honestly, I should definitely have a DJ. Alright. Hyperactive Ross protein issues signals on its own whether or not growth factor is bound to the... Okay. Alright, so the mutant is active without growth factor. Alright, this is worth remembering because 30% of human cancers are Ross. Which of the following would not typically cause an oncogene to become a proto-oncogene to become an oncogene? It's probably retroviral activation. This can move to a new control that's like always on or weirdly on. Gene suppression, if you knocked down a tumor suppressor gene, that's bad. Amplification, that's when the gene copies itself. And the point mutation, that could also do. So I'm going to say retroviral activation. Nah, I don't think it's gene suppression. Wait, is it? I don't think that's right. I wish she had the answers. Oh, wait. Bum, bum, bum. All right, so we need to go to, what week was this in? 8B. All right. Lecture recordings. I've never watched a lecture, but I just want to double check.
Okay, so we'll be talking. So that's the end checkpoint. That would halt that. Okay. As there actually isn't an S checkpoint that we talked about. You can see that it. So that again, those are normals. And certainly, and so this is platelet and whether or not factor dependence.
Whoa, the skipping in this is actually really good.
You see, so that's why.
This is like pretty nice. It's like better than YouTube.
Bottom here, so castrated attached to another two fat metabolisms again, and we'll look at these DNA mutation, which is abbreviated LOF for loss of function. You can see if they have either in a higher with depth control units. It's the growth so there are a lot of there's that. Remember we talked trigger. That makes sense. And again, that's trigger happening. So you can have confusion. P in to become an oncogene. So translocation can cause a proto effects. 2% of you chose gene suppression. That's probably the best answer. So again, the question is, which of the following would not typically cause a proto-oncogene to become an oncogene? So translocation can cause a proto-oncogene to become an oncogene because you could have the gene sequence under the control of a constitutive promoter, for instance.
Mm-hmm.
Turn it on all the time. Amplification is when you're making multiple copies of the gene. And so that can also, here's amplification, that can also result in excess expression of the protein. Okay. Point mutations can result in the rest hyperactivity or retroviral activation. Remember we talked about how viruses can result in cancer because they can integrate in a way that affects the control of other genes. Okay. So gene suppression would be the one that doesn't. If you're suppressing proto-oncogene, then you're decreasing the activity.
Oh, okay. So, okay, I get it. So what I was thinking of is just that it's like what leads to cancer, but it's like specifically what would not typically cause a proto-oncogene to become an oncogene, and it's gene suppression. If you turned off a proto-oncogene, but what is the definition of a proto-oncogene?
Just what you want.
What is it? Suppressed? Oh, it's 2P is suppressed. I think, okay, whatever. I'm not going to get hung up on it, but I feel like gene suppression is actually... Alright, whatever. So the answer is gene suppression. Okay. So there's two categories. The proto-oncogenes and the tumor suppressors. So, and so proto-genes push toward growth, and tumor suppressors inhibit growth. Okay? Normal genes function in delaying cell growth, locating or repairing damaged DNA, or initiating apoptosis. Normal and mutant cell cycle inhibiting pathway. Computer, make me more music like this. Whoa. That's actually crazy, man. There's a mutation that happens in 50% of human cancers. All right. UV light damaged protein kinases. Eukaryotes are so complicated. I mean, the fact that we have apoptosis as a thing is crazy. Which type of cancer-causing mutation is most likely to be recessive? What does that mean? Okay. It's got to be the okay, so then the answer is yeah, it's okay, so recessive. So I'm going to guess that it's loss of function mutation and tumor suppressor. Ooh. Nice. Transformation requires a series of mutation. Mutations in tumor suppressor gene allows cell divisions without adequate checks and repair of DNA damage. It's D. Actually, wait, where's all my... Indie? I feel like I have some more. Do I still have the filter on? Okay, good. No. Well. Oh, still sleepless. That's what I was thinking of. Can't sleep at night. No, I can't sleep. I can't sleep at night. Thinking about you lately. Having second thoughts about you, baby. And I can't sleep at night. No, I can't sleep. I can't sleep. I would say yes, right? The likelihood that that other cell is... That's what I thought. Sitting around thinking about you lately. Having second thoughts about you, baby. Okay. Oh. That was a terrible mix. But also, like, I don't need all that. Of two soft voices blended in perfection. Of this record that I found. You to me while I'm here, finding all my previous motives, growing increasingly unclear. This is what should have been doing all along. Lose some sales and my boss won't be happy, but there's only one thing on my mind. Searching boxes underneath the counters on a chance that I take my fine and fall. What am I supposed to know about this? It's like, okay. Alright, so we're just gonna study breast cancer, I guess. I just don't get the value of memorizing all this. Is there anything to learn? I mean, at this level. Obviously there is, at a deep level. Let's see. What does she say about this? What does she say about this?
Well, triggering the other. Oh. Think about the role of genetic screening again, please. We may have looking description right? And so that's a source of signaling as well. Okay. All right. So they've in that cancer genome project that I talked about earlier, they were studying breast cancer to try to see what types of genes are mutated, what types of biomarkers are there, can we categorize breast cancer into different types. So they developed. We're going to highlight 4 different types here. Okay. I'm going to sort of bin luminal A and luminal B together as sort of a category of breast cancer. What do we see? So again, remember, normal expression of these different receptors is 1+. We're seeing 3 pluses, 2 to 3 pluses for the estrogen receptor, 2 pluses for the progesterone receptor.
Okay, so luminal has.
That means we're seeing less than normal expression of HER2, then higher than normal expression of the progesterone receptor and the estrogen receptor. I've highlighted the estrogen receptor in red because one of the ways we can treat this type of breast cancer where you're overexpressing the estrogen receptor is to use treatments that specifically bind to the estrogen receptor. So one of those is tamoxifen. Tamoxifen acts as a competitive inhibitor for the estrogen receptor, so it competes with estrogen for binding the receptor. And if tamoxifen is bound instead of estrogen, then you don't get the downstream effects of the estrogen receptor activating other gene expression. Okay, so you're blocking the signal. So that's one of the therapies if you have a type of breast cancer that overexpresses the estrogen receptor. There are other therapies that can target the progesterone receptor if it's overexpressed. Okay, that makes sense. So 2 other subtypes of breast cancer. One is called basal neg or triple negative because you have lower than normal expression of the estrogen receptor and the progesterone receptor.
So this is triple negative.
And the HER2 receptor. That's why it's called triple negative, because all 3 of these receptors are being expressed at lower rates than normal, or lower levels than normal. Okay. So 15 to 20% of breast cancers fall into this triple negative or basal-like class of breast cancer. This is also typical of cancers caused by seen in patients that have BRCA1 or 2 mutation. Okay. Unfortunately, triple negative breast cancer is more aggressive than some of the other subtypes of breast cancer. And it's also harder to treat because you don't have overexpression of any of these receptors. So you can't target a receptor that's not there. Okay? So it can be harder to treat. So the tamoxifen that we used earlier when you have overexpression of the estrogen receptor won't work here because you don't have overexpression of the estrogen receptor. Does that make sense? So treatments that target any of these 3 receptors are usually not effective. This last class I want to look at a little bit because it overexpresses the HER2 receptor tyrosine kinase receptor. So you see much higher levels of the HER2 receptor than normal, sometimes 100 times higher than normal. Okay. That's not necessarily great, but it gives you a target for treatment. There is a target, or there is a treatment called Herceptin that was developed by Genentech. It's an antibody that specifically binds to the HER2 receptor. Okay, so let's see how that works. So HER2, again, is one of the subunits. It's one of the receptor tyrosine kinase subunits in this HER class of receptor tyrosine kinases. There's another variant called HER3. Okay, a signaling molecule, like a growth factor, can bind to HER3, and that will trigger dimerization between HER2 and the HER3 monomer. Okay, so HER2 doesn't have to bind to another HER2 monomer; it can bind to HER3. Once they dimerize, that activates things, right? Remember how receptor tyrosine kinases activate each other is they take ATP and phosphorylate the other subunit. So the HER2 monomer will phosphorylate the tyrosines on the HER3 monomer, and vice versa. The HER3 monomer will phosphorylate the tyrosines on the HER2 monomer. So now those tyrosines are fully phosphorylated. Now this whole complex is activated. And now all the relay proteins downstream of the receptor tyrosine kinase signal transduction pathway are activated. And the response is enhanced cell division. So you get increased division of the cancer cells. Okay? So again, HER2 is a receptor tyrosine kinase. It can dimerize with other receptor tyrosine kinases like HER3 to be activated so that you phosphorylate each other and then trigger the rest of the signal transduction. Okay? Cancer cells can express over 100 times more HER2 receptors than normal. So we can target those HER2 receptors that are abnormally expressed. We can help treat the cancer. Okay? So Herceptin is the treatment. Again, Herceptin is an antibody that specifically binds to the HER2 monomer. When it binds to the HER2 monomer, it blocks dimerization with HER3. Okay, and if you can't dimerize, you're not activated and you don't go on and trigger cell division, right? So Herceptin is an antibody that inactivates HER2 by blocking dimerization and signaling. Oops. And so that is a very promising treatment. Herceptin is a type of chemotherapy, but because it's so targeted, it specifically interacts only with HER2 that's overexpressed in the breast cancer cells. It has very much reduced side effect profile than some of the general types of chemotherapy. The general types of chemotherapy often target things like microtubules that are important for the spindle fibers in dividing cells, right? So that will affect cancer cells because they're dividing rapidly. However, it also affects normally dividing cells like epithelial cells. That's why there's so many side effects with many types of chemotherapy, like hair loss, because hair follicles are a subtype of epithelial cell. They divide frequently. You can have a lot of GI side effects because all the epithelial cells that line your intestinal tract and all your digestive tract are affected by a lot of the chemotherapy.
Yeah, man. Modern chemotherapy is like bloodletting. Shit is crazy.
It can be very, very helpful to treat cancer with fewer side effects than general chemotherapy.
That's crazy, man.
So, key takeaways.
I'm listening. Yes, I'm guilty, and you should know
it. That affect cell cycle control. So you can have mutations that make proto-oncogene products overly active, so that turns them into oncogenes. So that's like your stuck accelerator. So that's like the RAS protein being hyperactive and always stimulating cell division, even when there's no growth factor bound to the growth factor receptor. We also looked at mutations that reduce the activity of tumor suppressor genes. So that's like having broken brains, like P53. And so P53 usually triggered a pause in the cell cycle and triggered repair of DNA damage. It triggered programmed cell death or apoptosis if the DNA damage could not be repaired. It triggered a pause in the cell division of the cell cycle. But when you turn those brakes off, then you no longer have those checks and balances. Right? Cancer involves a multi-step accumulation of mutations. Genetic screening can be used to identify risk, determine cancer stage and prognosis, and effective treatment for that particular type of cancer. And then we looked at how genomics-based studies propose 4 different subtypes of breast cancer based on gene expression, and then therefore that can help tailor treatments depending on which subtype of cancer you have. Okay? So,
all right, cool. All right, I think we're good on this. Let me just look at the study guide. Oh, wait, where's the study guide? Wait, first I want to do this. Okay. So we're 2 lectures in. We have 1, 2, 3, 4, 5. 5. Yeah. Anchorage dependence. Okay, cells only divide if they're like connected to something. Density dependence. They inhibit growth when the density of cells is high enough. Senescence is planned death after a certain number of divisions. Transformation is the change of proto-oncogenes into oncogenes. Apoptosis is programmed cell death. Hyperplasia is when you have an abundance of cells. There's too many cells, but they're pretty much normal. Dysplasia is when you have too many cells, but they're starting to look a little funky. Tumor in situ is is just like a worse version of dysplasia. Malignancy is when it starts to migrate. Mutagen, that's okay. Carcinogen. Spontaneous mutation, induced mutation, loss of function, gain of function, proto-oncogene, oncogene, tumor suppressor, hereditary cancer, sporadic cancer, ER alpha, PR, HER2, Herceptin. I would say maybe just the end of those things. All right, let's do meiosis. Ooh.
Esto se jodió, la vecina no sé qué bebió, el vecino no sé qué prendió, a la gente no sé qué le dio, pero todo el mundo está loco, todo el mundo está loco, todo el mundo rayado del coco. Y yo solo sé que montaron. Se te notan los pilates, o tú ganas o yo gano, no lo dejamos en empate. En mi casa se remate. No fuimos low key, callados sin dar detalles. La gente ya se dio cuenta que montamos la disco en la calle. Y esto es en el barrio. Siempre hay bailoteo, Ave María. El trago nunca falta ni la buena compañía. Yeah, cómo ha cambiado la vida. Yo crecí en el gueto y el mundo en la casa mía.
sent them a location. I said, I paid for the vacation.
I can't ever show face shit. I sent them a location. Falling when it's silent, silent when I'm falling. Call it undecided. I might quit. Ain't no pain, I'm gonna die. In my wrist, in a lie, I won't spend all my money on some shit I don't want. Ain't no sense in the thought, in my chest, in a night, I won't stay where it's sunny. If I can't leave the block, ooh, run it up. Said enough, no, wait it up. Fuck it up, I'm on the bitches up. Lace the forces up for love. I feel the tension, dark and wish you wash the dirt, but my cuffs. Ride it up, give me some space. I'm sided up, can't feel in my face. I lined it up, pick up the pace. I tied it up, you ain't in the race. My partner, I'm running away. I tried it up, my feelings are real. I'm shining now, I'm feeling okay. I'll ride it out. Hold my butt on mission, if I got it, I'ma run it till I die. I'ma make my life a show that just to show them I'm alive. Intuition, I don't want it, I won't take my own advice. But oh no, handlebars, gaslight burning under stars. Ain't no pain, I'm gonna die in my wrist, in a lie, I won't spend all my money on some shit I don't want. Ain't no sense in the thought, in my chest, in a night, I won't stay where it's sunny. If I can't leave the block, I hate it, I hate it, I hate it. Thanks so much, I hate it, I hate it, I hate it. Thanks so much, I hate it, I hate it, I hate it. Thanks so much, I hate it, I hate it, I hate it, I hate it. Can't believe I thought I would want this. Throw away everything I own. Running laps and it right where I started. Burning that wooden bridge on the roof of my apartment.
I've been sitting around thinking about you lately. Having second thoughts about you, baby. And I can't sleep at night. I can't sleep at night.
Synaptonema. Will I see you again? Um, isn't there a way to like? Last night I had a dream about you. In this dream, I'm dancing right beside you, and it looked like everyone was having fun. The kind of feeling I waited so long. Oh, come a little closer till the rhythm gets stronger. All night long. Oh. Minus Swan. Or Metaphase, yeah. We were standing side by side. I don't cross the takes to come alive. Oh, it's the way I'm feeling. I just can't deny. Yeah, that it makes sense. This song just goes so. Damn, Rihanna's actually still so popular. That's actually nuts. What's her number? What? She's the number 4 artist in the world? Who's number 1? That's insane. "102, she's number 5. Rihanna is bigger than Taylor Swift. That's insane. Okay, who is number 1? I don't even know. Drake? I don't even think Drake's bigger than them. Who's the biggest artist in the world? Dude, I don't even know who it could be. Who is number 1? Bad Bunny maybe? I don't know. Played the Super Bowl. 113. So that's number 3. Dude, I feel like it's gonna be obvious when I. Dude, no way it's Bruno Mars. Is it really? Wow, that's amazing, man. That's crazy, dude. It's Bruno Mars. The Rihanna thing and the Bruno Mars, they're both kind of crazy. Okay, I'm satisfied with that.
Shit feels the same as it always has.
Know my name, but you know my past. Got these shades, I'll shatter glass.
Just to see you again, since you never came back. Fight back your tears and you're crying. You make a mistake and you hide it. Emotions show in your white eyes. You hide in your room till the sunrise. Fight back your tears and you're crying. You make a mistake and you hide it. Emotions show in your white eyes. You hide in your room till the sunrise. Just a couple days in the summer. You just say you touch, you got my number. If I can't swim, then it's my fault. Yeah, it's my bad that I'm awful. Sorry that it takes so long just for me to write a fucking song, but I can't help it. I can't help it. Stuck in my brain isn't healthy. Last time you told me that you never wanna see me again. And ever since then I've been waiting for the summer to end. I can't wait to make another mistake and hear about how I let you down. And I can't fucking explain, but it feels like every day with you not around.
All right, that one was chill because that one was really easy. Let's see. Let's look at the study guide.
I know I could have done better. I know I could have been hurt. I gotta say it's not over here. Not now. There's no use to go down. There's no use to suck around. There's no use to go down. It's a new day. So catch you up. Tears and you're crying. You make a mistake and you hide it. Emotions show in your white eyes.
All right, this one's a fun one, I think.
Fight back your tears and you're crying. You make a mistake and you hide it. Emotions show in your white eyes. You hide in your room till the sunrise. Fight back your tears and you're crying. You make a mistake and you hide it. Emotions show in your white eyes. You hide in your room till the sunrise. loosen sails and my boats won't be happy, but I can. Open water, just what you wanted. So don't leave me drowning. Don't let me down. Don't hold it back like that. Just pull me closer, baby. Oh, let's make the most of this before it's over. No time, don't wanna waste a feel like we can make it real. Don't wanna wait when you could be loving me. Signs, too busy reading minds. Already made it here. Don't overthink when you could be loving me. Get me started before this started. I could be giving someone all of me, totally. Don't hold it back like that. Just pull me closer, baby. Better make the most of this before it's over. Time, don't wanna waste a feel like we can make it real. Don't wanna wait when you could be loving me. Signs, too busy reading minds. Already made it here. Don't overthink when you could be loving me. Play it safe, case I don't feel the same. Oh, end of the day. Won't face it, you're too afraid to say it. Too intimidated when you could be loving me.
A plant of unknown genotype with tall stems and axial flowers.
Some sails and my boats won't be happy, but I can't stop listening to the sound of two soft voices, blended imperfection from the reels of this record that I found.
In the mirror asking me what I'm doing. Answer is B. Previous motives going increasingly unclear. Far all the riches I believe as soon as I have left
all the other options
There's no lyrics.
will wither in the light of my plan. So I lose some sails and my boss won't be happy, but there's only one thing on my mind. Searching boxes underneath the counter on a chance that on a tape I'd find a song for someone who needs somewhere to long for. Someone who needs somewhere to long for. Homesick, 'cause I no longer know where home is.
Imagine a cross of two triple heterozygous pea plants with tall stems and axial purple flowers. Determine what promotion it will have. Isn't it gonna be 1/4 times 1 3/4 squared? What is this even, what is this picture about? Why is that there?
It's sad to think, I guess neither one of us wants to be the first to say wants to be the first to say goodbye. It's sad to think,
Okay, so today we'll be reviewing meiosis. That's chapter 13 in your book.
Oh wait,
Okay. And but we'll also talk a little bit more about genetics. Also next. You think that side between the DNA sequence
wait. Was this in here?
dependings of genetic inheritance because we understand
Was this in here? I didn't get those notes.
that chromosomes. Okay, so we're going to just so that 50, and then we'll finish with chromosomes. Like in love, just in a fused DNA sequence, right? So a base pair substitution. That is sufficient for many you can model system specific. Okay, so you write yeast genes differently than you write esophageal genes, which is different than how you write lowercase. It can be a physicist. They have one double heard of him before in some biology class in the past. But he was a monk, an Augustinian monk. He lived in the mid 1800s, and he was Austrian, although where he lived became part of present day Czechoslovakia. Okay, and so he was a monk. He could have plants that always gave purple flowers and always gave white flowers, and then he could breed them together and see what it got. He was lucky in that all the traits that he picked were not linked on the same chromosome. Again, we'll talk more about linkage next week. Okay, and he's, which is the part that creates the pollen, was critical to him being able to make the conclusions he did. Was he quantified his results carefully? Okay, so these are some of the traits that he looked at. So round versus wrinkled seeds, yellow versus green seeds, purple versus white flowers, instead of changed pods, green or yellow pods, whether the flowers were axially there on the sides or terminally there on the tips of the branches, and whether some terminal or short. So those are a lot of traits that he looked at. And so, again, the anatomy of the flower was important to how he was able to do control process. Okay, so we look at flowers a little bit in 16, right? So flowers are hermaphroditic, meaning that you have the stamen, which is the part that creates the pollen. So that's the male part of the flower. And then you have the carpel, which has the ovaries and the ovules inside them, which generate the eggs, right? So both the female and the male parts are in one flower, right? So oftentimes pea flowers will self-pollinate, meaning that the pollen from the stamen will pollinate the carpel of the same flower. Okay, so that's self-fertilization. But he was also able to control the process. So he could cut the stamens off from a flower before they generate pollen, and then that would prevent the flowers from self-pollinating. Okay, and so then he could use a paintbrush and then take pollen from a white flower and then brush it on the stamen.
Dude, where were these notes?
Okay, and that's what he got. And when he did that, he saw that all of the offspring all had purple flowers. Okay. All right, so he did what are called monohybrid crosses to study single traits. Okay, hybrid means that you're generating offspring that have different genetic combinations. Okay, monohybrid means you're looking at one trait. Okay, so he started with true breeding plants. So a true breeding plant was a plant that had purple flowers, for instance, and then when you cross that purple flower plant with another purple flower plant from that same lineage, all of the offspring were always purple. Okay, so those are true breeding plants. You always get purple flowers, or you always get white flowers. Right? You never get purple and white. You always just get one phenotype. Okay. Today we know that those true breeding plants were homozygous for all of the genes that were being studied. Okay. And then in these monohybrid crosses, again, mono means single or one, he made it individual that differed by only one trait. So purple flowers versus white flowers, but he didn't have different color seeds or different color pods or different flowers. And so again, he took purple flowers, true breeding purple flowers, and crossed them with true breeding white flowers. Again, he would take his paintbrush and put pollen from one flower onto the carpel of the other flower, vice versa. He did do the reciprocal crosses to see if he would get the same thing either way he did the cross. Okay. In most of the case, he did see the same results in the reciprocal crosses. Next week we'll talk about cases where you don't get the same results. Okay. But in the offspring, so parental generation is so that's the full on check one. Again, from F1s to get just the different cross. So today we'll be looking more at Mendelian genetics. Again, this is chapter 14 in your textbook. We started looking at methods of experimental quantitative approach on Monday, and we'll continue looking at the heterozygous should have very good cross. We'll generate those. So far so good. Cross. Come back to grades. You have it. So let's swap and have of meiosis, then you can really understand the law of segregation and the law of independent assortment, and then figure out the genotypes of the. It's the probability of either or. Okay, of zygote. Okay.
I just want to get to this picture.
Okay. All right. So let's look at dominance relationships. So we've been looking all the examples.
What's up with that picture?
For complete dominance. So the heterozygote has the same phenotype as the homozygous dominant. Okay. So the heterozygous individual has purple flowers, and that purple is indistinguishable from the homozygous dominant genotype. Okay, so those are the examples we've been looking at so far. But how can you tell?
She didn't even talk about that weird ass picture.
Slow news day. Look into the camera and apologize for offending all the nappers. I am really sorry about that. And honestly, I got away from me. I got nothing against you.
All right. Come on, let's finish this one out. Then we have how many left? 1, 2, 3. All right. So I'm trying to finish this by tonight so that I don't have to study this ever again. At least in this capacity. What makes an allele recessive? If there's no functional pigment protein, the flower will be white. All right, here we go. I'm gonna write this down. Actually, complete dominance is normal. Incomplete dominance. There's gotta be pictures. Roan cattle result from incomplete dominance. One, two, one, right?
Sad to think
Hope it's put together, but how much, and so get like an extension of look.
sad to think
All right, so we've looked at incomplete dominance as an extension of Mendelian genetics. Another thing that we want to look at in different dominance relationships is codominance as well as multiple alleles. Okay. So you may have heard of the ABO blood group and blood types, right? So you can have type A, type B, type AB, and type O blood, right? So these are the different blood groups. And so getting the right blood type, if you need a blood transfusion, is critical in order not to have any bad consequences. Because if you're transfused with the wrong blood type, you can get blood clotting. Okay. So what is the mechanism that underlies that? So it all comes from having different carbohydrates on the surface of the red blood cells. Okay. And so if you have a type A blood, then you have type A carbohydrates. And so here are the different carbohydrates. So the triangles are type A carbohydrate, the circles are type B, and then some alleles give you none. Okay, so if you type B blood, you have type B carbohydrates. If you have type A and B blood, you have both. You see both type A carbohydrates and type B. So this is codominance. We're seeing both the A phenotype and the B phenotype. Okay, so it's not a mixture of it's not, you don't only see one or the other, you see both. And so that's why it's called codominance. Okay, so there are actually three different alleles, okay, in this ABO blood group system. There's an IA allele, there's an IB allele, and there's a little I allele. Okay. And so for type A blood, you can either be homozygous for the IA allele, or you can be heterozygous with the IA and the little I allele. Okay, those will still give you type A carbohydrates on the surface of your red blood cells and give you type A blood. Similarly, for type B blood, you can be homozygous for the IB allele or heterozygous for the IB and the little I allele. To be type A, you have to have one IA allele and one IB allele. Type O doesn't have any of either of the carbohydrates. It doesn't have the type A or the type B carbohydrates on the surface, and that's homozygous recessive, little I, little I. Okay? So you can see a number of different things in this example. We see an example of codominance. If you have the IA and the IB alleles together, you see both the A phenotype and the B phenotype manifested. We also have dominance, complete dominance. We see that the IA allele and the IB allele are dominant to the little I allele. Okay, so in this case, the heterozygote has the same phenotype as the homozygous dominant genotype does. Okay, so, and then we see multiple alleles. We have 3 alleles in this system, right? So we see complete dominance, codominance, and multiple alleles here. Okay, so there's a question: are the heterozygotes, are they incomplete dominance? No, they're you see the same phenotype in the heterozygote as you do in the homozygote, so that's complete dominance. Okay, if you saw an intermediate phenotype for the het, that would be incomplete dominance, but we don't see an intermediate phenotype. We see the same phenotype between the heterozygous genotype and the homozygous genotype. Okay, all right. So there's a question: if red blood cells are enucleated, how do you look at the genes alleles? So you have to look at the genotypes of the precursors, or if the cells before they lose the nucleus. So it's a process, right? They're not born without a nucleus; they lose the nucleus, and so in the differentiation process, so you have to look at the stage where they still have the nucleus to genotype them. Okay. All right. You good? You guys good with this scenario here? So again, IA and IB are dominant over little I, but they're codominant to each other. Okay. All right. Let's see how that's sticking. Okay. 71% chose B, 14% chose C, and then 7% each A. And E. Okay, so how do you get a type O? How do you get type O blood?
I think it's just O.
I think it's just oh. What the? Actually, low key, I'm liking these lectures.
What genotype does that have to be? It has to be little I, little I. That's the only genotype that gives you type I. Recessive sector. Okay, that's the recessive phenotype. But there are other cases where there's a different phenotype. Okay, so it really depends. So let's start with you. Okay, let's talk a little bit about pleiotropy. So pleiotropy is when a gene has multiple effects, multiple phenotypes at different levels. And so let's look at sickle cell. So we talked a little bit about sickle cell and 6A. And so depending on what level you're looking at, you can have many, many different phenotypes. And so this gene is considered to be pleiotropic. So if you have 2 copies of the sickle cell allele, then all the hemoglobin that this individual is making is abnormal. Okay, it's a sickle cell variety, and that causes sickling because the hemoglobin makes these aggregations of long fibers, and that distorts the cell shape. Okay, these sickle-shaped cells have a lot of different result in a lot of different symptoms and phenotypes downstream. It can cause breakdown of the red blood cells. They don't live as long. They can get clumped and clot. They can clog the capillaries. Clogging the capillaries can cause organ damage because the blood isn't flowing because of the clog in the vessel. Right? It can cause a lot of pain. It can cause heart failure. The breakdown of the red blood cells can cause anemia and weakness. You're not carrying enough oxygen. You can accumulate the sickled cells in the spleen. The spleen is the organ that usually gets damaged cells, and that can cause spleen damage. Okay, and then downstream of that, you get all these other manifestations resulting from the damage to the organs. Okay, at different levels, you see different dominance types. Okay, so at the molecular level, the sickle cell allele and the wild type allele are co-dominant. Okay, in that you're producing 2 different kinds of hemoglobin. And at the molecular level, if you're looking at the proteins that are made, you can see both kinds of hemoglobin in the blood. You can see the normal hemoglobin and the sickle cell hemoglobin. Okay, so on that level, you can see co-dominance. Okay. But then if you're looking at phenotypes, a heterozygous individual may have an intermediate phenotype to a homozygous individual, an individual that has homozygous for the sickle cell allele, and that's different than the phenotype for the wild type, homozygous normal. Okay, so you have an intermediate phenotype if you're heterozygous, so that could be considered incomplete dominance. Okay, so the dominance relationship varies depending on the level of the phenotype that you're looking at. Okay, does that make sense? All right. So here's a summary table from your textbook. So they're looking at complete dominance. The heterozygous phenotype is the same as the homozygous dominant phenotype. Okay. Incomplete dominance, you see an intermediate phenotype between the 2 homozygous phenotypes. Co-dominance, you see both phenotypes being expressed. Multiple alleles, like the ABO blood group, you have multiple alleles. In any individual, you can only have 2 of the alleles that are available, right? But you have multiple alleles in the population. Okay? And then pleiotropies are when gene affects multiple phenotypic characters. Okay? And also, you can also see different dominance relationships depending on what level of analysis you're looking at. Does that make sense? So, you guys good so far in extensions to Mendel?
So pleiotrope.
dominance?
Okay,
So far we've
interesting.
talked all about
Oh.
carbon phenotypes.
All right.
Now we're going to look at the interaction between 2 genes. Okay, and so one example is something called epistasis. That's when 2 genes impact the phenotype that you see because they interact with each other. Okay, so coat color in Labrador retrievers is a classic example. Okay, so Labrador retrievers can come in 3 basic colors. We have black Labrador retrievers, we have chocolate or brown labs, and then we have yellow or golden labs. Okay? So there are 2 genes that are interacting to produce the coat color in labs. Okay,
It's a product and a co-product, bro. Not that I even know what that means.
so there's a black coat color gene. And you have 2 alleles. The dominant allele gives the black coat color. The recessive allele gives a brown coat color because they're producing less melanin. But
The expression or non-expression of the epistatic gene. Oh, interesting. Epistatic. Okay, that's pretty cool.
But then there's a second gene called the E gene, and the E gene is responsible for whether or not the pigment gets deposited in the hair of the lab's fur. Okay, so if at the E gene you have a homozygous recessive genotype, that means no pigment is deposited, and the coat color defaults to yellow, regardless of what alleles you have at the B locus. Okay, so you could be homozygous for the dominant B locus that would normally give you a black coat color, but if that pigment can't be deposited at all into the hair, you're never going to see that black coat color. Okay, so even if you're homozygous for the black coat color dominant alleles, if you're homozygous recessive and can't deposit the pigment, you're going to be a yellow lab. Okay, so all of these yellow labs know that they're homozygous recessive at the pigment deposition E gene. Okay, and as long as you have at least one dominant allele of the E gene, that means you can deposit the pigment, and so then we see the black versus the chocolate labs. Okay, so this is epistasis, where the expression or non-expression of one gene suppresses the expression of the other. So in this case, it's the E gene. If it's homozygous recessive, it's suppressing the expression of the second B gene, which would normally determine coat color.
Flamey flake.
It's still independent of sorting because all four of these genotypes or combinations are equally likely. So there's still the E alleles and the B alleles are still assorting independently of each other.
Bro finally streamed. Dude, that makes me happy, man.
That makes sense?
I got sick. I got kind of sick and I lost some motivation, bro.
Question so far in season.
Man, flamey flake, dude.
Okay, let's put on this one.
All right. Consider the case of recessive epistasis laboratory. Okay, so It's 3 to 2, but I'm confused on how do I calculate it.
Okay, so we're okay. We're little B. What? B. So it's what you so all right. Let's now just briefly look at polygenetic inheritance. All of these other traits that we've talked about, that Mendel worked on, etc., most of them are determined by single genes. But we actually know that that's not, most genetics is not that simple. Most phenotypes are the product of many, many genes interacting with each other. Okay? And obviously that's much harder to study, and it's much harder to figure out because it's much more complicated. So in your book, they have this example about skin color, but even this is vastly simplified because there are literally hundreds of genes so far that have been identified in determining skin color. Okay, but here they're oversimplifying it and saying, okay, let's say that we have 3 genes out of the hundreds of genes that have been identified so far. We're focusing on 3 genes. And if you have a dominant allele, that counts for like 1 unit of color. So if you have 3 dominant alleles in the heterozygote, you have 3 units of color, that'll give you a certain shade of skin color. Okay. So the number of dominant alleles will determine the color, essentially. So if you have no dominant alleles, then you have the lightest color. If you have 6 dominant alleles, you have the darkest color. And then any possible combination in between, based on the number of dominant alleles, will give you an intermediate color. Does that make sense? So again, this is a highly simplified version or example that they're setting forth. But here they're trying to show that the phenotype can have a quantitative basis and that's why you have a continuum or a spectrum of phenotypes in the middle. Okay. So that's all I'm going to say about that. Again, this is a highly simplified version, but again, there are many genes that can be involved in producing phenotypes, and how those genes interact, how many dominant alleles you have, can set a gradation of a continuum of phenotypes. Okay. All right, and then the environment can have a big role. Okay. And you've probably heard of nature versus nurture. How much of our phenotypes are determined by our genetics? How much of it are determined by our environment? And the truth of the matter is, it's a combination of both. Your genotype interacts with the environment to produce the phenotype. Okay. And we see that clearly in certain types of phenotypes. So you may have seen Himalayan rabbits and also California rabbits. Siamese cats are similar. What do you notice of the coloration pattern of the black cats? So these are the tips of the ears and the nose and the feet and sometimes the tail. So these are the parts of the body in the rabbit that are cooler. Okay. And so you see a different expression of pigment in the cooler regions of the animal than you do in the warmer regions of the animal. And again, remember the animal, the cells in the ear are genetically identical to the cells in the trunk of the animal, right? So we're seeing differential gene expression, and part of that is based on temperature in this case. Okay. And then we see, I'm going to talk about the hydrangeas next. So these are the hydrangeas, these are flowers, these are genetically identical, but what's different is the type of soil they're being grown in. So the pH of the soil, whether it's acidic or basic, and also whether or not aluminum is available in the soil. If you have acidic soil, you'll get the more reds and pinks. If you have basic soil with aluminum in it, you'll get more of the blue purple colors. And again, these are genetically identical plants, but they're grown in very different environmental conditions, which affects the gene expression. Okay? And then these are Arctic foxes, and then the photoperiod determines the coloration. So in the wintertime, when the amount of sunlight during the day is shorter, then you get white fur. When the days are longer, and so you have a longer photoperiod, you get darker fur. Okay? And you can see that could be adaptive because of camouflage reasons. Right? So the environment actually plays a huge role in the expression of phenotypes. So even given a specific genotype, the environment has a huge role to play in how those phenotypes get expressed from a given genotype. Okay. All right, so far so good. So I want to end on pedigree analysis. So that's how we follow genetics in human families. Okay. And so in a pedigree, a square represents a male, a circle represents a female. If the symbol is shaded, that means that that individual has whatever trait you're following. A horizontal line between two individuals represents the mating. And then the offspring are depicted coming from the mating, right? There's a vertical line that shows where the next generation is, and then the offspring are listed in birth order. So this is the oldest child, so this is the eldest daughter, and then you get a son, you get two sons, and then you get the youngest son, okay? So we have 3 generations shown in this pedigree. The trait we're following here is the presence or absence of a widow's peak. And so that's on the hairline, you have this little peak here, that's called a widow's peak. And so it's a dominant, actually we're trying to determine if it's a dominant or recessive trait, but you can sort of see that here in the pedigree, okay? So this individual has the trait, and so you can see that the individuals that have the trait all have the capital W allele. So this is a dominant trait. Another clue that it's a dominant trait is you see the trait appearing in every generation, okay? And that you can have every affected individual has an affected parent, okay? So you never see it skipping generations, okay? And so that's the hallmark of a dominant allele, okay? So in these pedigrees, what I would suggest is that you pencil in genotypes for all the individuals that you can tell. So here we can't tell whether this individual is homozygous dominant or heterozygous. We just know that they show the trait. But because we know the trait is dominant, these are the 2 possible genotypes for this individual. Her sister, however, we do know her genotype because she does not show the trait, so she must be homozygous recessive. Okay? So pencil in all of the genotypes for all of the individuals on these pedigrees, and it'll help you follow the inheritance. Okay? All right, so here's a recessive trait. So this is a tree for cystic fibrosis, okay, which is a genetic disease that affects the lungs. Okay, it actually affects the chloride channel in the epithelium of the lung. And so in a recessive trait, as opposed to the dominant trait that we just had in the previous slide, you can see all these unaffected individuals, and then all of a sudden you see affected individuals. So you can see affected individuals arising from unaffected parents. So that's a hallmark of a recessive disease. So what does that mean about the genotypes of the parents? So these affected individuals are homozygous recessive. That means the parents have to be heterozygous because they have to each have a recessive allele that they can pass on to their offspring. Does that make sense? So in recessive traits, affected individuals can be children of 2 unaffected parents. But if you have 2 affected parents, all the children of the affected parents should be affected also because 2 affected parents have to be homozygous recessive. So they can only give a recessive allele to their children. Okay? And then going back to dominant, what do you see here? You see affected individuals in all of the generations. Okay? So affected children always have at least 1 affected parent. The trait shows it in every generation, that's just skipping a generation. 2 affected parents can produce unaffected children. Do I have an example of that? Not a very clear one. If the parents are homozygous. Okay. So some other things. So a slash in a pedigree means that this individual has died or passed away. When you have 2 lines between individuals in a mating, that means that they're related. Okay, so here these are distant cousins in this tree, and that's also the case here. These are distant cousins. Sometimes geneticists want to track those types of matings because you're more likely to see the recessive alleles come up in the next generation. Okay. All right, let's do a poll question. Okay, so for those of you who clicked in,
This is a fucked up pedigree, bro. Hey, yo. Three-fourths, right?
57% chose 3 quarters, 25% chose 1 quarter, 8% chose 1. Okay, so let's think about this. So we're looking at individual 3-6 and then 4-6. So that's this meeting here. And so the diamond means that they had 14 kids and they're not specified as whether they're boys or girls or whether they were affected or not. Okay. So but with this, when these 2
Yo, they had what? They had 14 kids?
individuals have
Dude.
children, what's the probability that children will have?
Hey, I know we're cousins,
So
but
the
you
first
want to
question
have
is, is the dominant
14
or
kids with me?
recessive trait, right? So again, you can see it every generation. It doesn't skip generations. Every affected child has an affected parent. So that tells you that it's probably dominant. Okay? So that means that the genotypes of these individuals could be either heterozygous or homozygous dominant. Okay, so let's suppose, for instance, that they're heterozygous. Oh, actually, it tells you that they're heterozygous, right? So what's the probability that the child would inherit the disease? So they need at least 1 dominant allele from either parent.
Oh, shoot. Wait, that's not what I want. I don't want to do. All right, cool. Let's see. 2 more lectures left. Oh, wait, no. 3 more lectures left. Shit. There's actually a lot more. The last 3 are going to be the hardest ones. Bum, 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 bum. Dun, 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 dun. Ooh. Dun, 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 dun.
Okay, so this morning we're going to look at the
What
chromosome
the
theory
what
of
the hell is going on here?
infection. So it's an extension of a lot of the genetic concepts we've been thinking about.
It's like a different
But
player.
we're going to look at some of the evidence.
It's like a different player.
So
We're gonna finish up the chromosome theory of inheritance. So
Is
this
it?
is chapter 15 in your book.
I literally can't tell. It is. It's a totally different player.
that led
This thing sucks. What is this? Why is there this big gap here?
we'll look at sex-linked or X-linked genes and how they segregate slightly differently than autosomes do. We'll look at linked genes, which are produced on the same chromosome. And then we'll also look at
And the arrow keys don't work. You gotta be kidding me.
because we're probably not going to finish the last two today, so we'll finish them on Wednesday. Right, so we want to look at how chromosomes seem to be understood as the bodies that carry Mendel's inheritance factors. Remember when Mendel set forth his laws of segregation, his laws of assortment, he was relying on the numerical ratios, but he didn't know anything about what the molecular mechanisms were underlying the separation of the heritable factors, and then the dependent assortment, right? He was using just the numerical ratios. And so Mendel again published his papers around 1865. And so it wasn't until about 40 years later that scientists were really starting to look at chromosomes as potential carriers of the hereditary condition. Chromo is from the root chroma, for color, and some is from some of the body. So these are colored bodies that across this were looking at. And they colored these to develop lots of stains to help increase resolution and discriminate different structures. And so scientists were looking at chromosome. They were able to describe how DNA fused and how these structures inside the nucleus look like they were fusing also. Okay? And so they concluded that the nuclei had to have the hereditary material. And again, they were seeing these long thread-like bodies, and sometimes depending on the stains they used, they were sorted. And they would call it fused cells, which are very much like you saw different stages of chromosome contraction and movement in the reproductive cells that you guys know in cell, right? And so the chromosome theory of inheritance really started to come together in the beginning of the 1900s. So Walter Sutton, as well as Theodore Berry, were scientists that were independently developing these concepts. And so they saw that chromosomes have moving around. They saw that the chromosomes have different sizes, different shapes, the second ones have different positions, and different family chromosomes. And so they knew that they were helpful. They also saw under the microscope when they were watching the chromosomes, they saw the chromosomes spread up and have separate. And so they saw that different chromosomes were contributed by different each parent when they were watching fertilization. And so then they also noticed that the parent chromosomes separated from each other during meiosis. And so all of the things that were postulated by Mendel's law of segregation and law of assortment were supported by the movement of these chromosomes that these microscopes were watching from the chromosomes. And so again, chromosomes enter growth segregation.
All right, Jesus, what am I doing? Let's get to. This is probably the one I liked the least. Okay. Chromosomal basis of inheritance. I think I'm just gonna watch the lecture, bro. I'm not in it.
Assortment, just like Mendel's hereditary factors do. And then, so it was later, Thomas Hunt Morgan, we'll look at some of his experiments coming up. He was the
Dude,
one
the arrow key's not working is
color
so
things
dumb.
to develop a lot of stains to help increase resolution and discriminate different structures under the microscope, right? And so scientists were looking at fertilization under the microscope. They were able to describe how gametes fuse and how these structures inside the nucleus looked like they were fusing also. Okay? And so they concluded that the nuclei had to have the hereditary material. And again, they were seeing these long thread-like bodies, and sometimes depending on the stains that they used, they would color. And they could follow them through cell division, very much like you saw different stages of chromosome compaction and movement in the onion root tube slides that you guys looked at in 6A, right? And so the chromosome theory of inheritance really started coming together in the beginning of the 1900s. So Walter Sutton, as well as Theodore Boveri, were scientists that were independently developing these concepts. And so they saw that chromosomes had individuality. They saw that the chromosomes had different sizes, different shapes. The centromeres may have been in different positions. They had different banding patterns when they were stained. And so they knew that they weren't all the same. They also saw under the microscope when they were watching these chromosome movements, they saw the chromosomes paired up and then separated. And so, and they saw that different chromosomes were contributed by different each parent when they were watching fertilization happen. And so then they also noticed that the paired chromosomes separated from each other during meiosis. And so all of the things that were postulated by Mendel's law of segregation and assortment were supported by the movement of these chromosomes that these microscopists were watching under the microscope. And so again, chromosomes undergo segregation and independent assortment, just like Mendel's hereditary factors do. And then, so it was later, Thomas Hunt Morgan, we'll look at some of these experiments coming up. He was the one who really made the more direct link between genes being chromosomes, these bodies that are moving out of the. So here we have some chromosomes. These yellow dots are labeling a specific gene. And you can see that these are the two homologous chromosomes and that they've been duplicated. So we have one dot in each sister chromatid that's lighting up for the specific gene that this fluorescent probe is calling. Okay. So now we certainly know that genes are in chromosomes and we understand the molecular basis of that. But again, in the early 1900s, they were still putting all the evidence together. And so just a review of Mendel's laws and the chromosomal basis. So here we're starting with the yellow round seeds crossed with green wrinkled seeds. So these are true breeding homozygous strains. The traits for the seed color and seed shape are on different chromosomes. And so in meiosis, when gametes are being made and the gametes are haploid, you see one chromosome carrying the allele for the color, seed shape, and the other chromosome carrying the allele for seed color. And same with the other parent. Okay, so that's the parental generation. Then in the F1 generation, in meiosis, we saw the mechanism for the law of segregation, where the alleles for each gene separate from each other. That's the basis of the homologous chromosomes separating from each other in anaphase 1 and meiosis 1. So now we only have one of the alleles for each of the traits, and then that's the sister chromatid separating from meiosis 2. Right? Independent assortment, we see that the alleles on non-homologous chromosomes are independent. So the alleles for seed shape line up at the metaphase plate independently of the alleles for seed color because they're on different homologous chromosome pairs. Right? So we can have different configurations at metaphase 1 that leads to different combinations of alleles for the two traits in the gametes. Okay? So that was a lot of independent assortment. Right? And so again, we can, based on the assortment of the genotypes of the gametes, we can make predictions for the offspring genotypic and phenotypic ratios. Right? So this is a dihybrid cross, and so we saw a 9:3, 3:1 phenotypic ratio. Right? That's good there. All right. So again, Thomas Hunt Morgan He established what became known as the flywheel. He was a scientist at Columbia University in the early 1900s. And he was the first person to really decide that he was going to pursue Persepolis, this little fruit fly, as a model of genetic organism. So he had to develop this model system from the ground up. So he spent years learning to breed them and just taking really careful observations under the microscope of different traits, eye color, wing shape, bristle shape, etc. And so he characterized all of that. It took him almost 2 years to find his first mutant. And his first mutant was this one. You can see white eyes. Most of the normal flies have red eyes. So that's the reason it's red. And so again, fruit flies is one to develop Drosophila as a model genetic system. They're a good genetic system for similar reasons to how peas were a good genetic system for Mendel to work on. They have a fairly short generation time, so in 2 weeks you can go from eggs to adults. They generate fairly large numbers of offspring, over 100 offspring per cross. One really helpful thing, especially when they were trying to make the connection between chromosomes and these heritable factors that Mendel postulated, was the fact that Drosophila only have 4 pairs of chromosomes, so they're fairly large. So they're fairly easy to follow under the microscope. Okay? And so some of the vocabulary, the terminology that's used, so the common allele that's common in the wild population is termed wild type. And that's denoted by plus. And so the wild type eye color is red because that's the most common eye color in the wild population. Okay? And so the non-wild type ones are usually mutants. And so they are resulting from some sort of mutation. And so it's referred to as mutants. So this is an odd color mutant. Okay? And so the way that genes in Drosophila are named are usually after the mutant phenotype. So because this mutant phenotype has white eyes, whatever gene that was responsible for this phenotype was referred to as the W gene, the white eye gene, okay, the gene that confers white eyes. Okay? Remember, the one type or the most common or normal variant is referred to as the plus. So the red eye phenotype is noted as W plus for the wild type allele. Again, plus means that it's the wild type allele. Without the plus means it's the mutant allele. Okay? You guys good with the definition so far? Okay, so once, again, after a couple years or Thomas and Morton was trying to find mutants and trying to find characters that vary between different individual lines, he finally got his white eye panel. So he crossed his white eye panel to a red eye form of the panel to get to that one generation. All of the offspring had red eyes. So what did he call the dominance relationship between the know that the wild type allele is dominant because we're seeing the wild type phenotype, red eyes in the F1 generation, right? And this F1 generation would presumably be heterozygotes, right? So the heterozygotes are the ones, the individuals you can determine dominance relationships in, right? Okay. And so then Morgan did the F1 monohybrid cross. So you took red-eyed females from this F1 generation and crossed them with red-eyed males from this F1 generation to see what he would get. And so that's this F1 F1. He saw a 3-to-1 red-eyed to white-eyed phenotypic ratio, which seems normal. And so if you looked a little bit more closely, he saw that all of the white-eyed F2 progeny were male. Okay, so that was not typical of what Mendel was seeing, right? And so he saw a very specific sex ratio. And so again, it's a 3-to-1 phenotypic ratio. You have 2 of red eyes to white eyes, but all the males were white. All the males were white, I should say. None of the females had white eyes. Okay? So he knew right away that something weird was going on, and somehow this white-eyed phenotype was related to the sex of the flock. And he was trying to figure out what the mechanism of that was. Okay? So he referred to this as sex-linked inheritance. And when you see sex-linked now, think of X-linked because most of the genes that are sex-linked are on the X chromosome. Okay? The Y chromosome actually has very, very few genes. All right, and so for sex-linked inheritance, so now we're looking at the chromosomes. Okay, so in the parental generation, we have 2 X chromosomes. So in flies, females have 2 X chromosomes. Males have 1 X and a Y. We'll talk a little bit more about sex differentiation coming up. But again, females have 2 X's and males have an X and a Y in Drosophila. Okay, so the females in the parental generation had 2 wild-type alleles, so they were true breeding for red eyes. The male only has 1 X chromosome, so he only has 1 white-eyed allele. Okay, but he has the mutant white-eyed allele, okay, because he has white eyes and then he has a Y crest. Okay, and so again, XX Drosophila female, XY male. And so in the F1 generation, the sperm from this white-eyed male, the gametes can either carry the X chromosome with the white allele, the mutant white allele, or they can have the Y chromosome. Okay, one or the other. The eggs all have the wild-type allele at the eye color locus because this female in the parental generation was homozygous wild-type. Right? So when you do your Punnett square, you can see that all the females are heterozygous. They have 1 wild-type allele and 1 white-eyed mutant allele. And then the males are what are referred to as hemizygous. Hemi means half. I think I have that here. So hemizygous, they only have 1 allele because they only have 1 X chromosome, right? So we can't call them homozygous. We can't call them heterozygous, so they're hemizygous. Right? The male is hemizygous for the wild-type allele because it receives this wild-type allele from the egg, okay? And then it gets the Y chromosome from the sperm. Right? So far, so good in the F1 generation. So the F1 generation, again, all of the progeny or the offspring had red eyes, and that's what we're seeing here. Because they all have 1 wild-type allele. Okay, that's good. Okay, then in the F2, we take this female, cross it to this male, set up a Punnett square, so we can get these 2 gametes from the female, these 2 gametes from the male. We do your Punnett square, we see 3-to-1 red-to-white ratio, and then we look at the sex distribution. These are females because they have 2 X chromosomes. These are males because they have an X and they received a Y in the sperm. And you can see only the males show up as white because the females that have the white mutant allele also have another X that carries the wild-type allele. That makes sense? And so this is the basis for the 3-to-1 ratio, but only males have the white phenotype. Okay? And so this was the first solid evidence that a specific gene was associated with a specific chromosome. And so again, Morgan postulated that the white-eyed gene was on the X chromosome, and all of the phenotypic ratios, like this one in the F2 generation, corroborated that hypothesis. Does that make sense? All right, let's do the first poll question. Okay, so for those of you who clicked in, we're pretty split.
I think this was the original cross, right? I'm pretty sure it's A then. It's A.
42% chose D, 33% chose A, 17% C, and 8% B. All right, let's think through. So the first thing that you really need to do when you're looking at a problem like this is you need to write out the genotypes of all the individuals, okay? And so again, practice association where you have the X and the superscript for the allele, okay? That will help you follow the females and the males, okay? And so if your homozygous red eye, that means you have two wild type alleles, so you have XW plus, XW plus. If you're a homozygous red eye female. If you're a white-eyed male, you have one X and one Y, and then the X has to have the mutant allele because it's white eye, right? If you're homozygous white-eyed female, then you have to have two copies of the white allele. If you are a red-eyed male, then you have a wild-type allele for the eye color and a Y to make you male. If you're heterozygous red-eyed female, then you have one wild-type allele, one mutant allele. And then if you're white-eyed male, again, you're XWY. And then if you're heterozygous red-eyed female, again, you have wild-type and mutant on the X's. And then a red-eyed male, XW plus Y. Okay? So again, write out all the genotypes. And then the question is, which of the following processes could not result in a white-eyed male? Okay? So if you go back here, so the males, where do the males get their X chromosomes from? They get their X chromosomes from the eggs. So the X chromosomes are coming from mom. They're male because they're receiving a Y chromosome from the sperm part. Okay? But the X chromosomes all come from mom. Okay? So whatever X chromosome mom is will be whatever the males show. Does that make sense? So if we look at all these genotypes, which one could not result in a white-eyed Drosophila male progeny, then we look at the females, right? All of these females have a white-eyed allele on at least one of the X chromosomes, so they could produce an egg that has a white-eyed allele to produce a white-eyed male. Because remember, the progeny are getting the Y from the sperm, and the X is coming from the egg from the mom, right? So A is the only one that can't result in a white-eyed male because you don't have any white alleles in the mom's X chromosome. Right? They're all wild type, so this would all be red-eyed males. Does that make sense? So A is the correct answer. That cannot result in a white-eyed Drosophila male offspring. Does that make sense? Questions about that? So there are a couple questions in the chat. So one is, are men also hemizygous for certain alleles? Yes, because in human males are also have one X chromosome and one Y chromosome, right? So they're hemizygous for all of the genes on the X chromosome. They only have one copy. Okay?
Hemizygous.
Is the wild type allele always dominant? No. Sometimes the wild type allele is recessive. And so we may see some cases of that in some of the problems, but it can be either. It can be dominant or recessive. And again, how do you tell whether the wild type is dominant or recessive? You have to look at the heterozygote, right, to see the dominance relationships. Okay. There's a question: is it possible to get mutations in the Y chromosomes? It is possible to get mutations in the Y chromosomes, just like any other chromosome, but it's less common in part because the Y chromosome is quite small and it has very few genes in it. Okay, we'll look at the Y chromosome for humans has only like 78 genes on it. I have 25,000 genes in the human genome. Okay, so very small number of people. Okay. All right, so let's talk a little bit about chromosomal sex determination. So just as it applies, you have two X's that you're female, and you have one X and one Y for male. Okay, that's similar at the surface level anyway for human sex determination. If you have two X's, you're female. If you have an X and Y, you're male. Okay? The mechanism is actually a little bit different. In humans, it's the presence of the Y that makes an individual have male sex characteristics. In Drosophila, it's actually not the presence of the Y that determines whether you're male, but it's the number of X chromosomes that determines whether you're male. So in Drosophila, having one X chromosome makes you male. Having two X chromosomes makes you female. In humans, it's
Wait, she said Drosophila.
Okay. So slightly different, even though the genotype looks similar. Okay. There's also an expo system.
Oh, I forgot the stupid fucking program.
So far, so good in the F1 generation. So the F1 generation, yeah.
That went back.
The progeny.
That went what?
In some organisms like chickens,
That went back like
it's the female that has the different sex
fucking
chromosomes.
20 minutes. Jesus. Whatever. School. CD exam 3. Make their vids. CD vids. What is it called? How do I update? UV update. I don't actually give a shit.
Chromosomes separated out, like just so a 3-to-1 for you know 42 females see female. Why? All right, so let's talk a little bit about chromosomal sex determination because we just said that flies, if you have two X's, that you're female. If you have one X and one Y, you're male. Okay, that's similar at the surface level anyway for human sex determination. If you have two X's, you're female. If you have an X and a Y, you're male. Okay, the mechanism is actually a little bit different. In humans, it's the presence of the Y that makes an individual have male sex characteristics. Okay. In Drosophila, it's actually not the presence of the Y that determines whether you're male, but it's the number of X chromosomes that determines whether you're male. So in Drosophila, having one X chromosome makes you male. Having two X chromosomes makes you female. In humans, it's again the presence of the Y chromosome that makes you male. Okay, so slightly different, even though the genotype looks similar. Okay, there's also an XO system. Some organisms, like grasshoppers and also C. elegans, only have one type of sex chromosome. And if you have two copies of the X, you're female. If you have one copy of the X, you're male. In some organisms, like chickens, it's the female that has the different sex chromosomes. So they call that the ZW system. And so you have two different sex chromosomes for female in chickens. If you have two of the same sex chromosome, then you're male. And then there's also a haplodiploid system. So haploid, diploid. So this is true in ants and bees. So females result from fertilized eggs, so they're diploid. Males actually result from unfertilized eggs, so they're haploid. Okay, so there's a lot of different mechanisms for sex determination in different types of organisms. Okay.
Yo, bees are weird. Bees and ants are so weird.
And so here is the human X chromosome and the Y chromosome. You can see the Y chromosome is quite a bit smaller than the X. Okay, and the X chromosome has a lot of genes on it that have nothing to do with sex or sex determination. And so males and females need those genes on the X chromosomes, regardless of, again, independently of sex determination. Okay. All right, so there's a question, what made them change from X and Y to Z and W labels? It's because they didn't want it to get confusing. Because ZW, again, the ZW system is a system where the female has two different chromosomes and the males have the same, two of the same sex chromosomes. So they didn't want to confuse this system with the XY system, so they gave them different letters. Okay. All right, so this is the X chromosome. It's a fairly large chromosome. Again, many genes on the X chromosome have nothing to do with sex characteristics or sex determination. Some of these genes, these are listed as diseases that are caused by mutations in some of the genes on the X. But you can see there's a lot of different genes that have nothing to do with sex or sex determination. Okay. So there are X-linked recessive disorders, and we're going to look at how those get inherited. Okay, so color blindness, red-green color blindness, is caused by mutation in one of the genes on the X chromosome, and so individuals with red-green color blindness can't discriminate certain colors because of it affects the photoreceptors in the retina. Duchenne muscular dystrophy is caused by a mutation on the X chromosome. It causes progressive muscle degeneration. And then hemophilia, there's actually a few different types of hemophilia. that can affect blood clotting factors, factor 8 and factor 9. Those are both coded for genes on the X chromosome, and so that can cause internal bleeding and other issues. So hemophilia was seen in a lot of the royal families in Europe because Queen Victoria was a carrier, and so she passed on the mutant allele for hemophilia to her daughter. And then there was a lot of intermarriage between royal families in Europe. We see the Tsar of Russia marrying one of Queen Victoria's granddaughters, etc. And so this hemophilia spread throughout the royal families in Europe. Okay. So the inheritance of X-linked recessive traits, this is how you can follow it. So let's start with the parental cross, where we have homozygous
Bruh.
dominant or wild type female mating with an affected male. So affected is colored in sort of the darker orange, and he has an allele that is a mutant allele. So he shows the mutant phenotype. Okay. So we can see Punnett square, the eggs all get a wild type dominant allele from the mother, and they all get a mutant allele from the father, or at least the X chromosomes, and then, or they get a Y from the father. Okay, so when you go through the Punnett square, you can see that all of the female progeny.
I feel like that makes sense.
So first, the carrier female. So first, we go to the F2 generation.
Yeah.
Okay, so then now we do a cross between these two. We do a cross between a carrier female and an affected hemizygous mutant male. Okay, then what we see. So
Hemizygous mutant male.
the heterozygous female has one egg that has a normal allele, half of the eggs have a mutant allele. In the sperm, half of them have the mutant allele on the X, the other half have the Y, right? You do your Punnett square, you can see half of the females are carriers, the other half of the females are affected. The males have
All right, come on.
wild type allele, and the mutant allele. The phenotype having one copy of a gene sometimes give a different phenotype than having two copies of a particular allele. Having phenotypically and genotypically from the autosomal traits. Okay. All right, so let's talk a little bit about dosage compensation. So remember when we were looking at incomplete dominance, we saw that having one copy of a gene sometimes give a different phenotype than having two copies of a particular allele, okay, because of dosage, okay? So when we have different numbers of X chromosomes between males and females, that leads to something called X chromosome dosage compensation. So remember, most of the genes on the X chromosomes have nothing to do with either sex characteristics or sex determination. They have to do with, photoreceptors in the retina or muscle cell function and stuff. So you need some way to equalize the gene dosage between males and females for all of those genes on the X chromosomes, okay? And so that's referred to as dosage compensation, okay? And there are different mechanisms for dosage compensation depending on the organism. So in Drosophila, so again, males have one X and one Y, right? Females have two X's. So what Drosophila males do is they upregulate transcription from the one X that they have. So they essentially double the transcription from that one X, so that it essentially matches the dosage of regular transcription from two X's in the female, okay? So that's how Drosophila match the X chromosome genes. The dosage is they double the transcription in the males off of a single X, okay? In C. elegans, they kind of do the opposite. So again, females have two X's and males have one. The females, actually they're hermaphrodites in C. elegans, but they have two X's. They downregulate the activity of the X chromosome genes. So they reduce the transcription off of both X chromosomes by about half, so that you end up with about the same gene dosage as the one X from the male that's being transcribed at a normal level, okay? So again, in Drosophila, they double the transcription of the one X in the male, and in C. elegans, they reduce the transcriptional level from the two X's in the hermaphrodites, okay? And then in mammals, they do something different. So in mammals, they inactivate one of the two X's in the female. So that becomes something called a bar bond. So that's called X chromosome inactivation. So there's different mechanisms for dosage compensation, again, depending on the organism, but they all have a mechanism to equalize the transcription from the X chromosome between males and females. Okay, because again, most of these X chromosome genes are required for normal.
I kind of wondered about that. That's pretty interesting.
Or sex characteristics. Okay. All right. So far, so good. All right. So let's look a little bit more at X inactivation. Again, this happens only in mammals. Okay. So in female mammals, one of the two X chromosomes is inactivated, and which of the two chromosomes is inactivated is actually random. Okay. And this happens during embryonic development, okay? And again, this is to equalize the gene dosage between the females and the males, okay? So females only have one functional X, similar to males now, because one of the X's in females has been inactivated, okay? But because the inactivation is random, that actually leads to some really interesting genetics in females, okay? So now let's look at this cat here. So let's say the cat has two different alleles for fur color on the X chromosome. One allele is for orange fur and one allele is for black fur, okay? And again, randomly, one of the two X chromosomes will be inactivated. So this happens in embryonic development, usually between the 8 to 10 cell stage, and it's finalized by the about 100 cell stage in embryonic development, okay? So about half of the cells have the chromosome with the orange fur allele inactivated. The other half of the cells have the other X chromosome with the black fur allele inactivated, okay? And so these cells go on to divide through mitosis to give rise to daughter cells. So you get these clonal patches of different fur colors on the female cat. You get patches of orange fur that descended from a cell that had the chromosome with the black fur allele inactivated. Then you get patches of black fur that descended from cells that had the chromosome with the orange fur allele inactivated. So that's why you get tortoiseshell cats or pelican cats, because you get patches of cells that have one or the other X chromosome inactivated. Okay, and so if a female is heterozygous for any gene that's on an X chromosome, she will be mosaic for that trait, meaning she will have patches of cells that may have express one allele from one X chromosome, and then she'll have patches of cells that express the other allele from the other X chromosome. Okay, so that gives rise to again some very interesting genetics. So there are a number of diseases or disorders based on gene mutations on the X chromosome. One of them here is called Alport syndrome. It's again an X-linked disorder. It's caused by mutations in one of the collagen genes. There are a number of different collagen genes, and this one happens to be on the X chromosome. Remember, collagen was important as a component in connective tissue and as a component of the extracellular matrix. Collagen is a protein that forms fibers in the extracellular matrix. You guys remember that? So if you're a male and you're heterozygous, because you only have one X chromosome, right? And if you have a mutant allele in this collagen gene, you have a severe phenotype because all of your cells have that mutant allele, right? And so you have a lot of issues if you're not making this collagen gene appropriately, okay? Some symptoms are blood in the urine, kidney problems, hearing loss, a bunch of others, okay? But so if you're male and you have the mutant allele, you have a very severe phenotype. If you're a female and you're heterozygous, your phenotype can vary depending on which X chromosome randomly got inactivated and what the relative proportion was in the specific tissues that are most affected by this disorder. So you can have heterozygous females that have moderate to severe phenotypes, but you can also have heterozygous females that have no phenotype or very weak phenotypes. And again, that's based on the random nature of X inactivation, and again, which cells of which tissues had the mutant allele expressed versus inactivated. Does that make sense?
That's pretty
Questions on
interesting.
X inactivation? All right, so let's now think about linked genes. Okay, so we were talking about sex-linked genes, genes that are on the X chromosome. Be careful when we talk about genetic linkage, we're talking about genes that are linked together on a chromosome. It doesn't have to be the X chromosome, it could be an autosome. Okay, so be careful. There's a difference between gene linkage and then X linkage. Okay, so just don't confuse those two. Okay, so here we see again our dihybrid cross. We start with our true breeding pea plants. One has the wild type alleles or the dominant alleles. One has the recessive alleles. We get our dihybrid, so heterozygous at each of these two loci. We can see which of those traits are dominant versus recessive because it's the heterozygote. Right? We have both alleles in the background. And then we had our two different hypotheses. If these alleles from the parent segregate independently of each other, so that was independent assortment, right? We have the combinations that we saw in the parents, the two dominant alleles and the two recessive alleles, but we also had the other combinations possible. So we had the parental gamete genotypes as well as the recombinant gamete genotypes, but we expected them in equal proportions, 25% each, because this is the hypothesis that they are assorting independently of each other, that they're on different chromosomes, and then the chromosome that has the Y allele ignores the chromosome that has the R allele, and the homologous chromosomes are aligning at metaphase 1 and meiosis 1. You guys remember that? So this is independent assortment. We predict equal proportions of all the 4 possible genotypes in the gametes, and then we get our 9 to 3 to 3 to 1 phenotypic ratio. Okay? So that was independent assortment. The alternative hypothesis was dependent assortment. So that means that the gametes and the parents stay together and they don't separate from each other. And so we only get 2 possible genotypes in the gametes because, again, they're the 2 parental configurations. Okay, then we predict a 3 to 1 phenotypic ratio. We never see these recombinant gamete genotypes. Okay, that's dependent assortment. Okay, so alleles of different genes on different chromosomes segregate independently of each other. That was independent assortment, right? So the question is, if genes are on the same chromosome, do they segregate dependently like this? Dependent assortment. Okay, so let's see. So we're going to look at linked genes now. Okay, so Morgan did more experiments, and so he was looking at 2 genes for 2 different traits. One trait was for body color, So wild type phenotype is sort of a gray body. The mutant phenotype is a black body. And then he was looking at wings. And so normal wings are fairly long and extend beyond the end of the abdomen on the fly. And the mutant, you see these really short wings. These are called vestigial wings. Okay. And so he had a wild type female that he crossed with homozygous mutant male. So we're talking about autosomal mutations here. They're not on the X chromosome. Okay. So they're on some autosome. So it's a homozygous mutant male. So he's got two copies of whichever autosome this is on. Okay. And then we get our F1 dihybrid. So this is a heterozygote. Okay. It's got one wild type allele from mom, one mutant allele from dad for each of these traits. Okay. So far so good. And this double heterozygote has a normal body color and normal wings. So that tells you that the mutant allele is recessive to the wild type allele, right? Because you can always tell dominance in the heterozygote. Okay. So far so good. All right. So all the F1s have gray bodies and normal wings, and then that tells you dominance. And this is also consistent with what Mendel would predict. And so now we're doing a test cross. We're not doing an F1 to get F2, but we're going to do a test cross. Okay. So we're taking our dihybrid. Remember, test crosses are when you cross to homozygous recessive. Okay. So if we have dependent assortment, this heterozygous female can only make one of two types of alleles that look like the parents. So she could have the two wild type alleles from this original homozygous wild type parent, or she could have the two mutant alleles from this homozygous mutant parent. Okay, those are the only two types of gametes that can be made if it's completely dependent assortment. Okay, and then the test cross, you're crossing to the double homozygous recessive mutant. Okay, so that only one type of gamete can result with the mutant alleles, right? So when you do your Punnett square, you see 50% of the offspring in this test cross are heterozygous because they have the wild type alleles from the egg and the mutant alleles.
All right, I'm gonna have to rewind. Whoa, what the fuck was that? Yeah, it's like Minecraft. The hell? am I doing? I hate this linked gene stuff.
From the sperm, from the homozygous recessive. 50% of the offspring are mutant because they get the mutant alleles from the egg. Okay, so this is the phenotypic and genotypic predictions for dependent assortment. Okay, and then we want to contrast that with what we predict for independent assortment, because the reason we're doing the test cross is to see, are we observing independent assortment or dependent assortment, right? So for independent assortment, we expect all of the possible gamete types, both the parental types and the recombinant types.
Yeah, okay, that makes sense.
Because again, we're assuming that the 50% we have genotype. Right? So our actual outcomes, so out of 2,300 progeny, so Morgan's students were counting a lot of flies, 2,300 flies counted, about 42% had the parental types of offspring, wild type or black vestigial, and then not 8 to 9% showed the recombinant or non-parental type of offspring.
All right, so what was the cross? The cross was B plus VG, VG. Okay, so we have this dihybrid, and then we cross it again with the homozygous double mutant. Yeah, and there's way less of
Wild
those.
type
Yeah.
body, but mutant vestigial wings or combinant inherited by independent wing shape genes, black and vestigial, are linked, meaning that they're on the same chromosome. Okay, so this again is the image from the current image. Okay, the black chromosome, seeing the parental. Okay, so we now know what the mechanism of this is, right? But let's look at this. So Morgan determined that the genes that are close together on the same chromosome do not assort independently; they are genetically linked. Okay, unlike genes that are either separate chromosomes or from the same chromosome do assort independently. We now know that crossing over is the mechanism that results in the recombination of linked genes. Okay, so we've talked about crossing over in prophase 1 and meiosis 1. Okay? And so that is the mechanism by which we get different combinations. But here we're looking at, here's our dihybrid female and our homozygous male in the test cross. Again, if we don't have crossing over, you're going to get one of the two chromosomes from the dihybrid female, right? And then the other chromosome comes from the test cross male. Okay? So these are the parental phenotypes, right? So the amount of recombination that you see is directly related to how far apart or close together the two genes are. The closer the genes are to each other, the less crossing over you observe, and then the lower the number of recombinant individuals you see in your cross. Okay. All right. So going back to this cross, this is parental generation, homozygous wild type, homozygous recessive. Our F1 is heterozygous. It gets one chromosome from the homozygous wild type parent. It gets the two mutant alleles from the double E mutant parent. And then test cross it with our between the non-sister chromosomes, right? So we can swap pieces, and then we can get exchanges of different alleles. So now this chromosome that's recombinant has a wild type body color allele, but a mutant vestigial allele for wings. And then this chromatid has the reverse. It has the mutant allele for the body color, but the wild type allele from wings after the crossing over between these two genes. Okay.
Wait, let's do the poll question then. All right, yeah, so I guess I'm gonna have to do this. In tomatoes, heterozygous. So it's 23. Yeah, that's what I thought. Okay.
So then when we go on to meiosis 2, we can see that these are the recombinant chromosomes that resulted from crossing over. These are the parental chromosomes that resulted from regular segregation, right? And then we get the alleles from the test cross male, and these are the different combinations we get. So far, so good. So we do see recombinants, but we see them less frequently than 25% of the time, right? So we can calculate the frequency of recombinants that we see, okay? So to do that, it's the number of recombinants observed, 206 plus 185, divided by the total number of progenies. And in this case, it's 391, divided by 2300 total progeny, times 100 to give you 17% recombination, okay? And so we can call that 17 linkage units or map units as far as genetic distance goes, okay? And this is on chromosome 2. So far, so good? Any questions on this? All right, let's do this one question. With the lower percent, what the book uses chromosomes, we're assuming that we're duplicated here, okay? So that we have this duplicated pair has the dominant alleles, this duplicated pair has the recessive alleles, okay? You can write it this way, where it's horizontal instead of vertical, and then you have a line for each chromosome, and then one chromosome has the 2 dominant alleles, the other chromosome has the 2 recessive alleles. Okay, so far so good on this notation versus this. And then we can get rid of one of the lines, okay, and then collapse it to just one single line, where above the line is one homologous chromosome, below the line is the other homologous chromosome. So one homologous chromosome has both dominant alleles, the other homologous chromosome has both recessive alleles, okay? So this alone is more useful than this because it shows you which alleles are on the different chromosomes. Again, this original notation does not indicate that, okay? Another way to write this, if you have to collapse it a little bit more, is you can have the slash diagonal. Before the slash represents one homologous chromosome, after the slash represents the other, okay? So this is what you'll see a lot of times when it's typed, just because it's easier than typing it this way. Okay, so far so good in the notation? All right, so these are 2 possible ways that you could be heterozygous at each of the different genes, right? You can have both dominant alleles on one chromosome and both recessive alleles on the other. Or you can have one dominant allele on one chromosome and one recessive allele and then vice versa on the other. These are both different configurations that will give you this genotype. And that's why the way of writing this genotype with no slash is very uninformative, because you cannot distinguish these 2 possibilities. These 2 possibilities will behave very differently in test cross, right? Because you get different gametes. In this